everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to discuss about various biological fluids which are found at the crime scene. The most common biological fluid which is found at the crime scene is blood. The blood which can identify using various tests, we can go for the various preliminary or confirmatory tests, and then we can confirm whether this blood belongs to animal or human, and then we can associate those blood samples to a particular individual using DNA profiling. But apart from blood, depending upon the crime scene, the type of crime scene, there can be another biological fluids or other biological fluids that are present at the crime scene and which can also help us in the identification of a particular individual, because these biological fluids, they contain biological material, various cells, and through which we can extract DNA and we can find or nab the criminal using this biological fluids. When we say the blood is the topmost biological fluid which is found at the crime scene, the most common biological fluid which is found in the cases of the sexual assaults is semen. So, this semen is important in the investigations where the sexual assault cases, and we can help prove a crime was committed and we can identify the perpetrator using the identification of the semen. When we say what is semen, it is a semi-fluid mixture of cells, enzyme and other organic and inorganic materials. The semen also consists of sperm cells which are the most important component, as these are specific to a particular semen. So, whenever this semen, as we know, the blood substance, the blood fluid which is found at the crime scene, it is red in color and it is readily visible to the eyes. But this biological fluid semen, this is color, the color is transparent and it is not visible to the eye. So, what to do if we know the case is a sexual assault case, or we have certain idea that there might be some sexual assault or rape is involved, then we have to look for the traces of semen on various areas, for example, bed sheets or other various areas where the traces of semen can be found. For that, we use a certain light, UV light, in which the particles, they will glow, and we will get to know that where are the traces of semen present. So, what is semen? The ejaculate volume in the males, it is around 2 to 6 ml, and each ml contains 100 to 150 million sperm cells per ml. So, the next question is whether the sperm count is almost similar in each and every male. So, we will say it depends upon various males. The normal human being, if we consider, the sperm cells which are present in 1 ml of semen is around 100 to 150 million sperm cells. But there may be certain factors. For example, if a person is being under some disease, under the influence of drugs or alcohol, or if a person has undergone the process of vasectomy, or with through any genetical congenital factors, there might be certain ways that the sperm count of a person is very low. So, the sperm cells, when we say, we call it as spermatozoa. The head of the sperm cells, they contains DNA, and the tail, it is for the movement. The acrosome is the part, the structure at the front of the head, and this is the part where the enzymes are there which are being helpful for the penetration of the egg, and that is how when this acrosome, it strikes the egg in the female body, then it produces further an embryo. So, when we see the sperm cell, the picture looks like the head and the tail. The tail is basically to help in the movement of the sperm, and the top, which we say the head, which we consider the bulb like structure, it has the acrosome, and it is the one which goes and which strikes the female egg. So, this semen can be found on clothing, skin, bedding, etc. So, if we say the color is mostly transparent, but it is more on the little bit yellowish side. So, we have to look for the alternative light sources, as I mentioned, to know about the fluorescence. So, if we take a UV light and if we close the other lights in the room, then the fluorescence will be there where the semen stains are present, either on the clothes or the person's skin or on the bed or any other area like the bedroom, washroom, etc. Now, whenever you reach a crime scene, firstly you have to identify whether the stains of semen are present or not. To identify that, we are using the ultraviolet light. We have to collect semen samples. If they are present on any cloth material, then we directly cut that cloth material and pack that. But if the semen sample, if there is on any material which cannot be collected or tore off, like it is present on the walls or it is present on any table chair, then we have to swab those samples. Then these samples are being transferred to the forensic science laboratory. For that, we have to confirm whether these are semen sample or not. We do some preliminary test. This preliminary test for semen is seminal acid phosphatase, or we also call it as SAP test. This is a preliminary test, and as we have read in the blood also that the preliminary test might give false positive results with other similar substances. So, we cannot confirmately say if a SAP test is positive that the semen is present. The chances are there that it is present, but it could also be a false positive test. So, what is SAP? Seminal acid phosphatase. These are basically a class of enzymes which are found in various living tissues. For example, mammalian liver to cauliflower stem juice. Semen contains very high levels of these enzymes, acid phosphatases, and as it is present in semen, we call these enzymes are seminal acid phosphatase. The level of these enzymes are higher until 40, till the male person reaches an age of 40, but as the person age decreases, the level of these enzymes also decreases in the person, and these level are not related to the sperm count. So, what test we conduct is Brantamin fast blue test. This is the test for SAP, and most importantly, if this sample is diluted to 500 times also, then the Brantamin fast blue test will also give color. That means, if I say, for example, a sexual assault or a rape has been conducted in someone's house and that person has washed their bed sheets and their bed covers, still after washing also these tests will be positive, will be there present, the enzymes will be present there, because even if the dilution factor is 500x, still the Brantamin fast blue test will be appearing. So, this is a color changing test and the SAP reacts with the substrate. Whenever we are going for the Brantamin fast blue test, we use a moist swab or filter paper which is rubbed over the stain, and the reagent containing substrate and chromogen is dropped on the swab or the paper. So, we can see in the picture that a swab is there and we have a filter paper which is mostly these two, any of these two can be used. Supposedly, you have a suspected place, a wall of the room where you have suspected that a semen sample is present. So, we will take a moist swab or the filter paper and then we swab that area or we swab it through the filter paper, and after that we will put our fast blue B dye, and this fast blue B dye, if it gives the purple color, then we can identify that the semen might be present. As it is a preliminary test, we cannot say confirmately that semen will be present. So, this purple color, if a fresh stain will be there, the strength of the color will be very high. Old stains, they produce a weak reaction. The other materials, for example, urine, vaginal secretion, saliva, sweat, they may also produce weak reaction. So, at the end, it is the experience of the analyst which will determine how to proceed further. If he feels that the semen is present, then we can go for the further confirmatory test. Like in blood, there are confirmatory tests of Takayama and Tickman. In the identification of semen, we have confirmatory test of Christmas tree stain and the prostate specific antigen. The Christmas tree stain is for the specific identification of sperm cells. So, I will say, if a person or a criminal has a very sperm low count, for example, he is suffering from a condition, for example, where the sperm count is very low Azoospermia we call it, or oligospermia we call it, where only 8 to 10 sperms are found in his semen, then these tests, Christmas tree stains test, cannot be applied on that semen sample. Then we have to go to prostate specific antigen test, which is an antigen antibody reaction test. So, usually Christmas tree stain test goes for those samples only where the sperms are present. Why we call it as Christmas tree stain? Because two types of stains are used, one is green in color and one is red in color. As the green and red color signifies the look of the Christmas tree, then we call it as a Christmas tree stain test. This staining helps in distinguishing the sperm cells from the other epithelial cells. What we do in this Christmas tree stain cell, we collect the portion of the stain on the moist swab or take cutting of stain. We agitate the swab in the small tube with water and then smear the water on the slide, and we stain it with the red and green dyes. So, when we will get to know if the red dye is there, then the sperm cells are stained, and the green dye is there, then the epithelial cells are there. If the Christmas tree test does not come positive, maybe that is one of the major reason that the sample is too old, that the sperm cells did not survive. So, either the dead sperm cells can also give certain dye reactions, but in some cases when the sperm cells are not there, then we have to go for PCA, or we also call as P30. It is a protein produced by the prostate gland, and it can confirm semen in SAP plus samples that do not contain semen. So, this test relies on the antigen and antibody reactions. Where we use the PSA, we will take the sample from the crime scene and we use the prostate specific antibodies, and if it shows a precipitation, then we confirm that PSA is present. So, we swab or cutting agitated in water, we drop this to the test well and the liquid moves through the result well. If the antibody is already bound to the membrane, then a visible line means PSA is present, and other lines are there which are controls, which shows that test was done properly. So, basically to start with, we have to go for the preliminary test, which is seminal acid phosphatase test, where we use brentamide fast blue B, and for the confirmatory test we either go for the Christmas tree test or we go for the prostate specific antigen test, which can confirm, and with 100% confirmation we can say that the semen is present at the crime scene. So, this is mostly used in the cases which involve sexual offences and making it necessary to examine exhibits for the presence of seminal stains. For example, if a female, she complains of a rape case, then a swab has been taken from her vaginal area and that has to be checked for the presence of semen. Nowadays, there are certain cases which are fake cases and the female claims that sexual assault has been conducted, but in the cases there is no sexual assault is being conducted. So, in such cases also we have to take swabs and to confirm whether there is presence of semen or not. So, going the step by step process, firstly we have to identify the semen, the location of the semen, and test to perform its identity, and secondly, when we have confirmed that the semen is present, the next question which arises is whose semen it is. Who is the person who has conducted the sexual assault or rape on that particular female? So, we have to look for the blood types and we can also go for the DNA analysis. Now, when we say yes, the answer is for the blood type analysis we can also go for the semen, saliva, etc. There is one term known as secretors. 80% of the world population are secretors. That means their blood type antigens, which we call as A, B, O and RH, they are very high in concentration in their body fluids. For example, saliva, semen, vaginal secretions and gastric juices. So, this have A and B antigens higher than the blood, and we can say with clarity that which blood group this person belongs to. So, from the semen, saliva and urine, we can also identify the blood group of a person, which can help us in locating, or we can help us in associating that particular semen sample to the person. So, for the location, the semen is often readily visible on a fabric because of the crusty appearance, and then we can locate it with the acid phosphatase color test, which can be detected using the fast flow B dye or fluorescence under the UV light. Other substances, as I have mentioned, which can give a false positive result, they are fruit juices, contraceptive creams and vaginal secretions. But none of these will react with the speed of seminal fluid. That means if a reaction which occurs gives a purple color within 30 seconds, that is also one of the confirmatory analysis that the semen is present. The other things will also give results, but that will take a lot of time to get to know that how much time it will take to produce that purple color. The next we have to go for is the microscopic examination of the semen. The microscopic examination involves the sperm analysis, where we have the spermatozoa, its telltale shape, its head, tail, etc., which is absolute indication of the presence of semen. The rapid stirring of a fabric stained water mixture will transfer a small amount of sperm into the water. A drop is then taken, dried, stained and looked at under the microscope. So, when you have a semen sample under the microscope, what do you expect? We expect that in the semen sample there will be various sperms which are going from here to there, moving using their tails. Yes, the microscopic slides which we have seen in the starting, that is a microscopic slides where we can see sperms travelling depending upon how old the sample is. So, but there are certain microscopic problems. Locating sperm with a microscope is not that easy because they are very brittle and dried can easily disintegrate. For example, the head can be at one point and the tail can go to the other point. They are also hard to remove from the cloth material, and the sexual crimes, they involve males who have an abnormally low sperm count. For example, if a person is having oligospermia or azoospermia, then they have a low sperm count and there will not be any sperms present in their semen. So, in such cases we cannot go for the microscopic analysis. So, detecting semen, the major confirmatory are the P330 prostate specific antigen test and the Christmas stain test. So, at the crime scene, when we collect the rape evidences, the outer and undergarments are placed in separate paper bags. This is one point which has to be noted, that the female undergarments and the male undergarments, they have to be packed separately in the different paper bags. Another important thing which we have to keep in the mind, all the biological fluids, they have to be in the non-airtight containers, because in the airtight containers they might grow off certain microbial substances. So, they have to be collected in the proper paper bags. While victim stands on a piece of paper and the paper is then folded and collected. The beddings should taken, if a particular place we have located using the fluorescence light, then that area should be cut and taken, and the rape victim should be subjected to a medical examination. Collected items including pubic hairs, vaginal swabs, blood, fingernail scrapings, clothing and urine. Suspect must also undergo an exam. Routine items including the clothing, hairs, penile swab and blood samples should be conducted. A living or motile sperm may survive around 4 to 6 hours in the vagina of a living person, and a non-motile sperm may be found in a living female for up to 3 days and occasionally up to 6 days if the person or the female has not taken a bath. Intact sperm are not normally found 16 hours, but have been found as late as 72 hours even after the intercourse. So, what this considerations tells, that how long ago was the person being raped, when did voluntary sexual activity last occur prior to the sexual assault, and blood is taken from any consensual partner having sex with the victim within the 72 hours of the assault. So, that will help us in narrowing the person, and for further confirmation we can also go for the DNA analysis, which can be extracted from the semen samples. Now, the another biological fluid which is found at the crime scene is the saliva. Saliva is made up of water, mucus, enzymes and other stuff, and the humans produce around 1 to 1.5 liter of saliva in a day, which aids in digestion by lubricating food, and it begins breakdown of starches. So, how do we identify the saliva? The saliva contains an enzyme which we know as the amylase. This enzyme breaks down the starch. So, it is found in many body fluids, very high in saliva. Two tests we use is the starch iodine test and Fidibas reagent test. So, we know when iodine and starch they come together, they turns blue, and if we put the saliva sample over there, the amylase will break down the starch and the color will be subsided, that means blue color will turn into almost dark brown or black color. So, that is one of the preliminary tests we use for the saliva identification. So, we will take a gel containing starch which is stained with blue with iodine, sample is added, and if the amylase or saliva is present, the blue color begins to vanish, which will tell us confirmatively that a saliva is present in the sample. The other test which we is for saliva which we do is the starch linked to the color dye. The two methods we use in Fidibas reagent is press test and tube test, where the Fidibas reagent tablets dissolved in water sprayed on filter paper and allowed to dry. This paper placed on area to be tested, and paper is sprayed with water and pressed against the area where we have that saliva might be present. This paper is left on the area and washed for blue color change and can be washed for up to 40 minutes for the color change. In the tube test, what we do, we contain stain in the placed in tube and the Fidibas tablet is being added, centrifuged, and the amount of color in top liquid is measured by a spectrophotometer. So, this will also tell us about the how much quantity of saliva is present. If we have to identify that this saliva belongs to which person, if a person is secretor, then firstly we can go for the ABO antigen analysis, and if for the DNA analysis with some biological stains, DNA testing can be done. The cells which are present, the buccal cells which are present there, they can be taken and through that DNA can be extracted. So, most importantly, the saliva on the crime scene might be present if the person is involved in some kind of drinking, eating, etc., or maybe the person is a smoker and the cigarette butts are there. In such cases, that can be found. Another important biological fluid is urine, which is mostly found in the cases of harassment, mischief, sexual assault, and the stains can identify through the visual examination using the UV light, and they are rarely used, difficult to identify as urine and difficult to get a DNA profile using the urine samples. Mostly whenever there are the cases of mischiefs, when a person has used their urine samples, then we can identify those, or maybe in the cases of secretors they can be used and they can be indicated for the presence of the blood samples or to get to know whether which blood group this person belongs to. To confirm this is a urine sample, we look for urea or creatinine. These are in other body fluids also, but they are in lower concentration. So, we can get to know if the sample has a urine or not, but this is still a preliminary test. It is difficult to identify DNA from the urine, but this is not impossible. There might be some epithelial cells from the urinary tract lining and other cells. In stains, the cells are spread out, therefore, the typing is difficult. And as urine also composed of various microbial substances like bacteria, which makes the typing difficult. The cells may be concentrated from liquid samples, then the DNA typing is possible. So, mostly this could be present in the cases of harassment, sexual cases. There, in such cases, the urine samples can be present at the crime scene. They can be taken as swab, or they can be taken if a large amount of sample is present, then they can be taken using some form of injection and then can be sent to the forensic science laboratory using some plastic tubes or maybe swabs in the paper bags. Another important major biological material are the feces and the vomit. They can be found maybe same way as in the cases of the mischief or in the cases of sexual assaultment, or if there is any cases where the feces may be found at the crime scene. Mostly the fecal identification goes to the wildlife analysis, the wildlife forensics. This fecal material are the cases come usually from the wildlife, where in the wildlife sanctuaries we have to identify which animal is present, or if any animal is being attacked by another animal, or the pattern of various or the poaching pattern we have to note. In such cases the fecal matter mostly come from the cases of the animals or the wildlife. But in case if the fecal materials are present, they can be present for the humans also. Feces are basically the end product after the digestion. This undigested food, mucosal cells, bacteria, they are being found in the feces. They are identified by the greenish brown color odor undigested food, and depending upon in the cases where for the medical reasons also, sometimes the stool samples or the fecal samples, they are being sent to the pathological laboratories where they can identify if the person is suffering from any particular medical disorder or disease depending upon the fecal material. They can be test for urobilinogen, but this stain must be apparent. So, depending upon what kind of cases are there, these fecal material, depending upon what kind of question is being asked by the field investigator or is being asked by the person who is present at the crime scene, and what kind of answer is he looking for or the person is looking for, then the same can be identified or the same fecal material has to be analyzed in such way. So, for the DNA testing of feces, low cell count is there and high bacteria, which makes it really difficult, but it is not impossible for that. So, to conclude, I will say all the biological materials, whether present in the small quantity or the high quantity at the crime scene, they have to be collected properly, sent properly to the lab. Only then a proper lab report can come, which can help us in solving the cases and submitting the report to the court of law for better judgment and analysis. Thank you.